Now, welcome to the session on uric acid metabolism. What I have done for this session is that I have uploaded two decks of slides. The first slide deck introduces you to the concepts underlying the metabolism of purines, and the second slide deck elaborates on the biochemistry associated with the pathophysiology of gout. Now, you may already know some of the concepts that are elaborated in slide deck 1, because these concepts most likely would have been covered in the course of molecular biology and principles of genetics. So if you are conversant with these concepts, you can skip slide deck 1 and directly start with slide deck 2 to look at the biochemistry underlying the pathophysiology of gout. What we are going to do in slide deck 1 is to understand the general structure of purines, then look at the broad concepts associated with the catabolism of purines. And when we are talking about the catabolism of purines, I would like to talk a bit about the purine salvage pathway, which is important to understand some of the disease states which are mentioned in slide deck 2. So that particular part will be covered towards the end of slide deck 1. Now, before I talk about the catabolism of purines, let us try to define what is a nucleotide. Now, a nucleotide, which you may already know, consists of three components. First, it consists of a heterocyclic base, which is shown here in the figure. Then it consists of a sugar, which consists of 5 carbons. That is, it's a pentose sugar, which is shown here in the slide. And then the third component in the nucleotide is a phosphate group. So a nucleotide simply consists of a heterocyclic base, a pentose sugar, and a phosphate group. Now, a purine nucleotide consists of a heterocyclic purine base, and if I focus on the structure of purines, you will see that this is consisting of a pyrimidine ring, which is this particular part of the purine base, and this particular pyrimidine ring is fused to an imidazole ring. Therefore, purines are heterocyclic aromatic compounds consisting of a pyrimidine ring which is fused to an imidazole ring. Now, each component of the purine is derived from specific molecules. So let us quickly visit them. This particular carbon here at the 6th position is derived from carbon dioxide. This particular nitrogen at position 1 is derived from the amino acid aspartate. these two nitrogens at positions 3 and 9 are derived from the amino acid glutamine. This particular nitrogen at position 7 is derived from glycine, and this particular carbon here at position 8 as well as the carbon at position 2 is derived from tetrahydrofolate. And this is important because compounds that inhibit formation of tetrahydrofolate therefore block purine synthesis, and these compounds are therefore sometimes used as anti-cancer drugs. Now there are two purine bases that you would have heard about. One is adenine and the other one is guanine, and both adenine and guanine are present in the nucleic acids that you had been introduced to in the course of molecular biology and principles of genetics. So this slide actually kind of recaps what is a nucleotide, the structure of a purine base, and also identifies the different compounds from which the different atoms in a purine base is derived. So let us move on. How is purine synthesized in our body? Now you have to understand that there are three processes that contribute to purine nucleotide biosynthesis, and one of the most important processes is the synthesis of purines from amphibolic intermediates, which is also known as the de novo synthesis of purine nucleotides. Now, in the purine nucleotide, you need a pentose sugar, and this particular sugar is derived from ribose 5-phosphate, and this ribose 5-phosphate is derived from the hexose monophosphate shunt or the pentose phosphate pathway. And I believe we talked about this in the course of biochemistry. The ribose 5-phosphate is converted to phosphoribosyl pyrophosphate, also known as PRPP in short, and the enzyme that catalyzes this conversion is phosphoribosyl pyrophosphate synthetase. And this particular step is the rate limiting step for purine and pyrimidine biosynthesis, and pyrimidine happens to be the other heterocyclic aromatic base that is present in nucleic acids. However, in this particular session we will focus only on the synthesis and catabolism of purine bases. Now, once I have phosphoribosyl pyrophosphate in the presence of the enzyme glutamine phosphoribosyl pyrophosphate amidotransferase, it is converted to 5-phosphoribosyl amine, and through successive steps this is converted to inosine monophosphate, and these successive steps require the different molecules that give rise to the different part of the purine ring, which I mentioned previously. Once we have inosine monophosphate, it is converted to adenosine monophosphate and guanosine monophosphate through several successive reactions. I'm not going to focus on these reactions because for your course, these reactions are not important. So once we have adenosine monophosphate and guanosine monophosphate, we have the basic purine nucleotides which are used by the body for various functions. Now, when we try to understand the metabolism of uric acid, we have to focus mostly on the catabolism of purines, because the catabolism of purine produces uric acid. In short, in case of humans and higher primates, adenosine and guanosine, these two purine bases, are catabolized to uric acid. And if I want to understand the process of catabolism very simply, what is happening is adenosine is first converted to inosine by the enzyme adenosine deaminase, and inosine is converted to hypoxanthine and then to xanthine. Similarly, guanosine is converted to xanthine through guanine, and xanthine is then oxidized to uric acid by the enzyme xanthine oxidase. Now, this particular enzyme we talked a bit when we studied enzyme inhibition back in biochemistry, because the drug that inhibits this particular enzyme also attenuates the formation of uric acid and therefore is used for the treatment of gout. So if I want to quickly go through the catabolism of purines again, we have adenosine, which is converted to inosine, and you can see here inosine is converted to hypoxanthine, and hypoxanthine is then converted to xanthine, and xanthine to uric acid. Guanosine, on the other hand, is converted to guanine, guanine to xanthine, and xanthine to uric acid. So that's like a very simple outline involving the catabolism of purine that leads to the formation of uric acid. Now, as I mentioned previously, xanthine oxidase is the enzyme that is responsible for catalyzing the conversion of xanthine to uric acid, and this particular enzyme is therefore a target for drugs that are used for attenuating the formation of uric acid or inhibiting the formation of uric acid. Now, xanthine oxidase is predominantly found in the hepatocytes and is a metalloflavoprotein which contains flavin adenine dinucleotide and molybdenum and iron. Now, it is said that anemia is a risk factor for gout. In anemia, we have anomalous iron metabolism. It is not known how anemia leads to gout, but there is a prevalent thought that in anemia there is an alteration in the structure of xanthine oxidase which leads to gout-like symptoms. Also, the enzyme catalyzed by xanthine oxidase produces reactive hydrogen species in the form of hydrogen peroxide, and this hydrogen peroxide is converted to water and oxygen by the enzyme catalase, because superoxide radicals have to be neutralized in the physiological environment. Now, this particular article here, you may want to look at this, talks about or tries to map the correlation between anemia and the onset of gout. Although I have to say that the article doesn't elaborate much on the mechanism that is associated with gout-like symptoms in patients who are suffering from anemia. Now, why uric acid that is produced at the end of purine catabolism poses a problem in humans and higher primates? The key reason behind this is that uric acid has very low solubility, and at lower temperatures this uric acid crystallizes, and this particular phenomena is responsible for gout-like symptoms. Now, in other animals other than humans and primates, this uric acid is converted by an enzyme uricase to a metabolite known as allantoin, and this is the structure of the enzyme uricase. Why it is important, we will quickly talk about it in slide deck 2, but this particular enzyme converts uric acid to a metabolite known as allantoin, and this allantoin has high solubility. However, this particular enzyme is not present in humans, and therefore uric acid, if not eliminated at lower temperatures, crystallizes, leading to gout-like symptoms. So, what are the adaptations in humans that facilitate the elimination of uric acid, or that more precisely counter the uric acid problem? Now, so far, what you can see is the end product of purine catabolism is uric acid. Now, if you look at the structure of uric acid, let us quickly go back to one of the previous slides. You will see that it contains a lot of nitrogen atoms. Now, one way that the humans have countered the uric acid problem is that nitrogen is not excreted as uric acid in humans. In case of humans, nitrogen is converted to urea, and the process that is involved in the conversion of nitrogen to urea is the urea cycle, and we have talked about this in the course of biochemistry. Since nitrogen is not excreted as uric acid in humans, therefore the production of uric acid is comparatively low in humans than other animals who excrete nitrogen in the form of uric acid. Since humans convert nitrogen to urea, humans and other higher primates are categorized as ureotelic animals. Now, if I go back to the catabolism of uric acid, we see here that at various steps we are converting guanosine to guanine and adenosine to inosine. And what human beings do, or what happens in the physiological environment of humans, is that not all of this hypoxanthine and guanine is excreted in the form of uric acid, but part of it is salvaged. That means part of these metabolites are converted to inosine monophosphate and guanosine monophosphate, IMP and GMP, through a separate pathway known as the purine salvage pathway. So human beings are able to salvage part of the metabolites which are intermediates in the formation of uric acid. As a result, the levels of uric acid in humans are pretty low. So through these two mechanisms, human beings are able to counter the uric acid problem. So if I want to understand what I just said quickly, we have purines, adenosine and guanosine. Adenosine is converted to hypoxanthine through the intermediate inosine. Guanosine converted to guanine, and what happens is part of the hypoxanthine and part of the guanine is salvaged, as a result of which the level of uric acid that is produced, or the quantities of uric acid that is produced, is relatively low, and therefore human beings are able to counter the problem of low solubility and augmented deposition probability of uric acid. Now, in order for the salvage reaction to proceed, we need a particular enzyme. Most of the time in an assessment, they will mention the abbreviated form of the enzyme, which is AGPRT, which stands for hypoxanthine guanine phosphoribosyl transferase. And this particular enzyme is able to salvage part of the intermediate metabolites, that is hypoxanthine and guanine, attenuating the levels of uric acid which is produced by the catabolism of purines. So if you are able to understand these broader concepts associated with the metabolism of purines, I believe this will be enough for your assessment as well as for your understanding of the problems associated with uric acid metabolism. And therefore, what we will now do is to proceed to slide deck 2, where we will talk about clinical correlations which are more important for your purpose. As I mentioned previously, if you are conversant with these concepts, you may opt for directly looking at slide deck 2. But I believe it will be good for you to listen to this particular lecture because it will facilitate a better understanding when we talk about the clinical correlations. So I'm going to end the session here and move on to slide deck 2, which talks about the biochemistry underlying the pathophysiology associated with uric acid metabolism. Thank you very much.